Good afternoon. So if you haven't gotten your exams, Esther has them, right? So after class you can get them. You don't want to take them now because you won't pay attention to me. So afterwards. Today we're going to start talking about infection of organisms, not just cells. So we're going to look at some of the principles of pathogenesis. The rest of the course will deal with this aspect of how viruses infect you. So we're going to talk about infections in the real world, not in a culture dish. And this actually refers to part 3 of the tripartite strategy, which we talked about way in the beginning. That is, all viral genomes are able to establish themselves in a host population so that the viruses can keep going on and on and on. And I would suggest that you have a look at Appendix B in the textbook. Appendix B is in Volume 2. It's called Pathogenicity Snapshots, and it gives you an overview of individual viral diseases. I'll be mentioning various viral diseases from time to time, and it will help if you've got some familiarity with them. It's mostly a visual appendix, so I think that would be very useful. It has outlines of the viruses, the epidemiology, and the disease mechanism. So, some basic facts. Every host today, that's us and other animals, plants, insects that are hosts for viruses, has intrinsic defenses and immune defenses, and we will talk about how they differ later on, that have evolved to deal with infections, viral infections, bacterial, fungal, all sorts, as well as tumors. And every virus has to counter those defenses, otherwise there wouldn't be any viruses around. We will talk a lot about this, modulation of host defenses. This is the interface of virus-host interaction. It's a very interesting area of study. This is one of my favorite lines. We live and prosper in a literal cloud of viruses. If you could stain them, you'd see them floating around in the air here. But as we said before, most infections are inconsequential. Most of the particles floating in this room, which were left from the previous class, they will never find a living cell to infect. They'll land on your skin, which of course is dead, and they can't multiply them there. They will be inactivated by drying or ultraviolet light. Not much here now, but in the summer there will be. And none of them get beyond 1 or 2 cells in the initial site of infection. If we do happen to get infected, and I'll bet we're all infected on a daily basis, these little bursts of infection, a few cells here and there, but we don't know about it. If we do get infected beyond that, we don't have symptoms. Many infections are what we call inapparent. This is an important concept. The vast majority of most infections don't cause any symptoms that you can recognize. So you think you're fine and there's nothing happening, but there really is. Virus in these cases could be replicating and being transmitted. So even though you don't feel ill, you could be transmitting virus to others. It's likely that you are, in fact. Here's an example, West Nile virus infection. The West Nile virus was not present in the U.S. Before, what year? Don't remember. Somebody have a textbook with them? No. I can't remember the year. But it came to the U.S. very late, in the late 90s perhaps. Dick Despommier will tell you all about this at the end of the course. By October 2004, not many years after it was introduced, by the way, it probably came from an airplane from Israel, but we're not sure about that. Came to the U.S. in New York City in the summer. By October 2004, about 1 million people were infected. We know this because they did serosurveys for the presence of antibodies. Only 20% of the people had fever, and only 1% of the people had neuroinvasion, which is good, because when you get neuroinvasion, it's not good. Many people were infected with no obvious disease with West Nile virus during that period, and they continue to be. And this leads to transmission because you give blood and you don't know you're infected. If you give an organ, you can give someone virus because they don't check organs for every possible virus. They probably should, but not yet. And it makes it hard to stop an epidemic because you can't recognize it early enough. because people are shedding viruses and they don't have symptoms. If you have symptoms, you take care of them, but if you don't, you transmit the infection. So, two facts and a paradox. All hosts have remarkable defenses that block most viral infections before they start. This should be clear by now. We have lots of infections and we are healthy because we can take care of them. But some infections aren't stopped. We get sick and many die of viral infections. So what happened to the defense system? Where did they go? And how can a virus with a couple of genes even for the smallest virus, how can a virus make us sick and kill us? So these are some of the questions we'll try to answer in the rest of this course. And we'll be talking about viral pathogenesis, and pathogenesis is the process of producing a disease. We talk about viral pathogenesis, it is produced by a virus, but it could be bacterial or fungal or protozoan as well. And viral disease, that is when you get sick with a virus infection, you have a viral disease if you have symptoms. It is a combination of the effects of the virus replicating in the host and the effects of the host response on both the virus and yourself. So I want you to forget the idea that the virus is doing everything to make you sick. You are contributing by having an immune response against the virus. Virus infections span from benign, which we've been talking about, inapparent infections. They go from there to lethal. That's quite a range, and everything in between. There can be acute infections that are over very quickly, and there can be persistent infections that go on for years and years. And different mechanisms are at play, and we'll talk about some of those. A little history. For many, many years of our existence, we didn't understand about microbes as infectious agents. We thought when we got sick, it was because the air was poisoned and we breathed it in and it had toxins in it and it made us sick. And that's why there were epidemics of contagious disease. We knew there were epidemics of disease and that people spread it, but we didn't know anything about microbes. That association didn't happen until the late 1800s, when Robert Koch said microorganisms, which had been brought to the world by Louis Pasteur, he said these can cause disease, and he formulated his Koch's postulates to identify the agent responsible for a specific disease. He coined the word a pathogen and said microbes can be pathogens. So it's really not a very long time that we have this understanding of microbes as infectious agents, but we have learned a lot. Here is a wonderful picture of three of the main players in yellow fever. Yellow fever virus was the first human virus to be identified in 1901. And three of the major players in this story were Carlos Finlay, Jesse Lazier, and Walter Reed. And they showed that a virus was the cause of yellow fever. They were interested in this because they're trying to build a canal through Panama, and mosquitoes were transmitting disease to a lot of the workers. So this is a painting showing these three gentlemen here, and they've got a tube full of mosquitoes, and they are letting them bite one of these individuals here to show that you can transmit yellow fever with a mosquito. So this, of course, was not an IRB-approved experiment. In fact, in 1901, there was no IRB. You can't do this. This is why we use animals nowadays. You can't experiment on humans. But, in fact, over the years, many scientists did experiment on themselves. Some of the questions that we'll be tackling for the rest of the course. How does a virus enter a host? Well, how does the host respond? Where does the virus replicate, at least initially? How does the virus spread? What kinds of organs does it infect? Doesn't always infect every organ. And if it doesn't, why? Is the infection cleared right away? Is it a quick or acute infection? Or do you establish a persistent infection that goes on for years and years? And finally, how do you transmit the infection to other hosts? It's not always the same for all viruses. To have a successful infection, there are a number of requirements. You have to have enough viruses. You have to be infected with enough viruses, and that differs according to the virus and the particular situation. It may be a few particles, it may be a few million particles. The cells where the virus lands have to be accessible. They have to be susceptible and permissive. Those last two terms, of course, you know. We've talked about those before. but accessible, the virus has to be able to reach them. If it can't, there won't be a successful infection. The local antiviral defenses have to be at least initially defective, sometimes absent entirely, in order to allow the virus to replicate. So how do you get enough viruses at the site of infections? How many viruses does it take to infect a host? And that varies for every virus and for every host. It may vary for one virus and all of us in this room. It may take different numbers of the same virus to infect all of us. Would you like to try an experiment? No, we can't do that. Many variables conspire to complicate the issue. Host genetics, we're all outbred, of course. Host antiviral defenses, viral virulence, the particular gene composition of the virus may make a difference, the social behavior of the hosts, the age, and even the weather and the environment can make a difference. For example, the natural environment is a considerable barrier to infection. These are very small viruses for the most part. Remember, you can't see them. They're tiny, being blown around by air currents. So once they're released from the host, they face huge obstacles. There's a big dilution effect. So effective multiplicities when you are infected are quite low, very different from a cell culture dish. Sunlight is bad for viruses. The UV light will inactivate the genome. It will also dry it out. And a lot of viruses can't take that. The chemical environment can also reduce the numbers of infectious viruses. Many viruses are sensitive to heat, drying, and sunlight. And many viruses overcome this obstacle by making a lot of progeny. And they figure if I make billions of viruses, a few will get through and infect the host. So this is one of the selective forces for making large numbers of progeny. Humans, of course, don't have the same approach. We make very few because we try to ensure that we all survive. Many viruses are also stable to low pH or proteases. And those are the ones that can last in the environment on cooking surfaces in a cruise ship, for example, and so that you can transmit it on to other people. These are particularly viruses that are passed in the alimentary tract, that are transmitted by fecal-oral transmission, and they're waterborne infections. Some viruses never experience the environment. The life cycles involve insect vectors. The mosquito or some other vector bites you and delivers the virus right into your cells, so that virus doesn't have to deal with the environment at all. It multiplies in you, and then it's picked up by another insect vector and transmitted to another person. And some infections are spread by physical contact, of course, transferred by body fluids, and in that case, the viruses are not outside for long. So there are these are all different ways that viruses can be transmitted. And the viruses have evolved to be effective by their ways. So a virus transferred by an insect vector would not be stable to sunlight, ultraviolet light, and drying. But many respiratory viruses have evolved to be relatively stable under those conditions. The getting into the host is a crucial part of initiating infection. And as humans, we have a limited spectrum of sites for viral infection. We are mostly covered with skin, and of course the outer layer is dead, so viruses cannot replicate in the skin. If you just put viruses on your skin, they will sit there. Unless you have a cut or some kind of abrasion, a virus will not be able to replicate. So we're pretty well protected. The problem is we have these mucosal surfaces. We have this respiratory and alimentary tract. We have eyes, urogenital tracts. These have nicely exposed epithelial surfaces which are not protected as well as the skin, and that's where most of the virus infections initiate. And of course we need to have mucosal surfaces because we have to exchange material with the environment. We have to respire and expire and eat food and expel it and so forth. So it's a property of our evolution that we have these susceptible surfaces, and that's where viruses come in. So here is a diagram of some of the places that viruses come in. The eyes, we have nice mucous membranes there. The respiratory, alimentary, and urogenital tracts also. And even though the skin is protected, vector bites can get viruses beneath the dead outer layer, as can scratches, needle sticks, dog bites, etc. So there are ways to get past the skin. The skin is a good barrier, as I've said. Unless broken up, it's impervious to infection, but we do that regularly, as you know, and so we do get a lot of infections in the skin. We have the surface being dead shells, and those that land on the skin can be inactivated by drying. The skin is rather acidic. And there are other, we now are learning that there is a whole microbiome, bacteria and other organisms living on our skin, and many of those secrete compounds that protect us, interestingly. We don't totally understand this yet, but there are many bacteria on all of us that probably are barriers to infection with pathogens. Normal skin has a low pH. So the outer layer, the epidermis, the top layer, is dead, and you cannot infect it, as we said, unless you puncture it. But many ways to do that: skin abrasions, insect or animal bites, needle punctures. Once you get below the epidermis, then you have lots of blood vessels and lymph vessels, and the viruses that can get in there then can spread to other sites. So if you infect the very upper layers, there is not a lot of vascularization and transport there. Not easy for viruses to spread, but if they can get to the underlying layers, the dermis, then they can spread to other tissues. Mucosal surfaces are terrific places to initiate a viral infection. It's a wet, moist environment, so there's no drying out, not much UV light for the most part. And there are some other primary defenses that protect them, but they are nice vulnerable sites. Let's take a look at the respiratory tract. This turns out to be the most common route of virus entry. Why? Huge absorptive area, 140 square meters in your lungs. It's amazing. 6 liters per minute of a ventilation rate, a lot of alveoli, so you're always sampling the air. 6 liters a minute is a lot. And so, as I said, the air is full of viruses, so the tract, the respiratory tract, is a good place to introduce them. There are lots of replicating cells, and you have easy access to the blood from the respiratory tract. And typically we get viral infections by aerosolized droplets. Someone is coughing or sneezing or just talking. You know, in talking you expel a lot of droplets. And if I had a virus infection, if I had a measles virus infection, you would all be getting measles. Of course, you're all immunized, so you wouldn't be doing that nowadays. But in the old days, before immunization, measles, person standing in front of the room with measles would infect everyone from these little droplets going out and carrying viruses. In healthy people, the respiratory tract defenses are strong. We have mucus, 20 to 200 milliliters a day of mucus. Of course, you don't see most of it because it remains in your tract. It's good that you don't see most of it. I don't want to see most of your 200 mLs of mucus either. But you have a lot of mucus, and that's an important defense because it traps pathogens, viruses as well as bacteria, spores. And if you inhale these particles and they get down into your lower tract, the mucus is brought back up by ciliary action, and then you swallow it. Isn't it wonderful? You swallow the mucus and you inactivate all these respiratory viruses. And that you do that whether you like it or not. You are always swallowing this. The mucociliary escalator, we call it. It moves the mucus from lungs to esophagus at 1 centimeter a minute. So now you know what is going on as you're sitting there listening to me. You also filter particles out in your sinuses to a certain extent. So in addition to these physical methods, we also have immune cells in our lower tract. We have macrophages and other kinds of cells. We have antibodies if you have been previously infected. So we do have some good protection, and that is why we stay healthy. So here is a typical respiratory tract going from the upper to the lower tract. It is lined with a epithelial cell layer shown here, nice brush border epithelial cells interspersed with goblet cells that produce mucus, and these epithelial cells are ciliated, and that pushes the mucus up towards your esophagus so that you can swallow it. Viruses can replicate throughout the tract. They can replicate from the upper to the lower tract, and when they do so, and you have symptoms, they have different names, like rhinitis, the common cold, the very upper tract infections, pharyngitis or laryngitis, and all the way down to tracheitis, bronchitis, bronchiolitis, and bronchopneumonia. And some viruses can replicate at both sites and cause various syndromes, and others are restricted. On this slide, which is somewhat outdated now, we see rhinovirus is a upper tract pathogen. This is what we thought for many years, but now there is new evidence that in fact rhinoviruses can also cause pneumonia, which of course is an infection of the lower tract as well. The alimentary tract is also a wonderful place to meet new viruses. Eating, drinking, and other social activities routinely introduce viruses there. The alimentary tract is designed to digest food, right? It's always in motion, especially after you eat. Good opportunity for a virus to interact with a cell. And that's what, of course, the virus has to do. It has to attach to a cell lining the gut tract. So this movement provides that opportunity. The gut, though, is more hostile than the respiratory tract. The stomach is acidic. The intestine is alkaline. We have enzymes, bile detergents, mucus, antibodies, phagocytic cells everywhere. So this is part of the reason why we stay healthy. Nevertheless, viruses can go through all of this. Poliovirus you can ingest. It will pass all the way through your stomach and small intestine and replicate in your small intestine. There are even viruses that require proteases in your gut to activate them for infection. Those are some of the reoviruses. We'll talk a little bit about some of those wonderful gastroenteritis viruses later. Those are the reoviruses and the noroviruses that can pass through your gut. Gut's also lined with a epithelial layer. You have villi, of course, and the villi are composed of layers or sheets of epithelial cells magnified up here, and those are called enterocytes. Some viruses can multiply in the enterocytes. There are also specialized cells interspersed among the enterocytes. They're called M cells. These are involved in sampling the antigenic content of the lumen of the gut and saying whether there are bad things moving around that we should be taking notice of. So you can view the intestinal tract very much like the lung as a selectively permeable barrier. It needs to take in food that you have eaten, of course. So viruses take advantage of that. And because we have these M cells sampling the antigen content of the lumen, there are immune cells right below them. You see lymphocytes and macrophages very close to the M cell. Here is, in fact, an electron micrograph of an M cell. This is a section of gut. Here's the M cell, and here are the enterocytes on either side, lymphocytes and macrophages underneath. So the function of those cells are to patrol the M cell. The M cell is bringing in material from the gut. They're very active at transcytosis. They take in material here, they put it below to these antigen sampling cells, and they look at it and say whether it's a problem or not. But viruses exploit that. Here is, in fact, infection of an M cell by reoviruses, these little particles here. Reoviruses have exploited this uptake mechanism, this transcytosis by which viruses go from the lumen into the underlying tissues. The urogenital tract is also a site of infection. It is protected by mucus and low pH, but abrasions from sexual activity may allow viruses to get across the membrane there. So these membranes are not as well developed at taking up material like the respiratory and the alimentary tract is, but abrasions allow viruses to enter. Viruses like human papillomavirus may produce local lesions when they infect there. Others can infect the urogenital tract and then spread to other sites like HIV and herpes simplex virus. The eye is also a route of entry. The sclera and the conjunctiva, typically infected by viruses. You blink every few seconds, and as you do, you wash away foreign particles. And that's a wonderful defense, because any virus that lands on your eye is immediately washed away and therefore has little opportunity to infect. But if you get some grit in your eye and you rub it and you get little microabrasions on your eye surface, then that will let virus particles in. So typically infection occurs after some kind of injury, grit, ophthalmological procedures, not using sterilized instruments, or hot tubs or swimming pools that aren't properly sanitized. Many examples of adenovirus eye infections from people going in a hot tub where there wasn't enough chlorine, and then over the next few days everybody gets red eyes like this one. It's a subconjunctival bleed caused by a viral infection. So a localized infection of the eye is called conjunctivitis, and some viruses produce this wonderful red eye, which is very scary, but it's for the most part harmless, goes away. Some viruses can infect the eye and spread. Enterovirus 70, which can cause conjunctivitis, can in some cases spread back, we think, via the optic nerve into the brain and cause paralysis. Herpes simplex virus can infect the cornea and can cause blindness. The virus can also spread to sensory ganglia. So virus infections of the eye are not trivial. So here's a summary of all the different routes of virus entry in those. You should be familiar with just the general ways that virus can get in, all these pathways that I've talked about, how a virus might enter the respiratory tract, the alimentary tract, for example. And as we talk about these viruses, you'll remember that poliovirus and rotaviruses infect via the alimentary tract. You'll remember that HIV can infect via the urogenital tract as well. Okay, so you are infected by any of these routes, the skin, respiratory, alimentary, urogenital, etc. And we've talked about just how they are open to virus infection. Let's now talk about how viruses spread from those initial sites. All right? Now, some viruses don't spread. Some of them replicate wherever they come in. So here we have a gut epithelium, and a virus can infect these enterocytes and just spread throughout the enterocytes and never go underneath and spread to the rest of the organism. So the virus spreads within the epithelium, but it is contained by the tissue structure and the immune system. So what I mean by that, the tissue structure below these epithelial layers is what is called a basement membrane. This is not really a membrane, but a structure made up of a variety of proteins and proteoglycans. But it is a barrier to routine penetration of the subepithelial tissues by microorganisms. So down here below the epithelium is the subepithelial tissue, and there are blood vessels and lymph vessels there. And so if you got there, you could pretty much go anywhere in the organism. So the structure of this is such to prevent that. So some viruses, in fact, are blocked by that. There are also some immune responses at the surface that will prevent further infection. But other viruses can spread beyond this primary site. Again, it could be a respiratory epithelium, alimentary tract, doesn't really matter. When that happens, we say that these are disseminated infections. And when many organs are infected, we call it a systemic infection. So it's, again, that's spreading from the primary site to other places. Two ways of describing that. In order to do that, the virus must breach this physical barrier as well as immune barriers to get into the subepithelial tissues. So there is the basement membrane, and its integrity, as you can see in this picture, can be compromised by inflammation and destruction. So inflammation, of course, is the recruitment of immune cells and the production of cytokines in an area, and these can disrupt the basement membrane. So the intent is good. The immune cells want to get rid of infection, but in doing so, they're causing damage, and that allows viruses to pass into the subepithelial tissues. Some viruses, of course, can destroy both the cell and the basement membrane just by replicating in them, and so that allows easy passage. But again, not every virus that infects these sites can do these things. It's quite selective. So the subepithelial tissues are the key to a disseminated infection. The virus encounters fluids, lymphatic system, and phagocytes. And the phagocytes, of course, are there to try and clear the infection, but they could pick up a virus and bring it somewhere else, inadvertently spreading the infection. So that's how you come to get into other tissues from one of these epithelial barriers, by penetrating the basement membrane and then spreading beyond it. Now, these are all polarized cells, of course. They have an apical, a basolateral domain, and there are also lateral domains between the cells. And viruses, it turns out, are produced sometimes at one of the two surfaces, the apical or basolateral, or both. And I want to show you an experiment that illustrates how important it is where a virus is produced to the pathogenesis of that infection. So this is a couple of electron micrographs illustrating apical and basolateral production. Remember, viruses can be produced by budding from the cell membrane. In a culture dish, these cells are not polarized. They just have one membrane, and the viruses all bud from the top. But in our bodies, cells are polarized. And here's influenza virus budding from the apical domain of a polarized epithelial cell. Here's measles virus budding from the apical domain. So if this were your respiratory tract, this is great, right? Because the virus is budding into the lumen, and you cough and you spread it. If the virus was budding just in the basolateral side, coughing wouldn't spread the infection to others. So these viruses have evolved to be transmitted by the respiratory route by virtue, in part, of their being produced at the apical domain. Now, vesicular stomatitis virus is produced at the basolateral domain of polarized epithelial cells. This is not typically a virus that is spread in a respiratory route. These are, in animals, can be spread by vectors. It's typically an animal infection, and among animals usually spread by vectors. So it doesn't need to be spread in an apical way. Now, so basolateral release provides access to underlying tissues. Remember, the basement membrane is here below this epithelial sheet, and if the viruses bud down, they have potentially the ability to get past the basement membrane. If they bud just from the top of the cell, they're not going to get below it. There's no way that's going to happen. But that is good for other purposes, of course. So here's a nice example of how you can manipulate this and change the pathogenesis. Sendai virus is a negative-stranded enveloped virus. You can infect mice with Sendai virus, and the virus is released from the apical domain of the respiratory tract. And if you infect mice with a wild-type virus, it causes a respiratory tract infection, and it never spreads to any other tissues. But you can make a mutant of the virus by introducing a couple of amino acid changes in the virus, and that mutant is now released from both the apical and the basolateral domains of the cell. So what do you think happens when you infect mice with that mutant? Well, the virus spreads systemically. It then infects multiple organs and is lethal to the mouse. So the simple attribute of being able to bud from the basolateral domain of a cell enables a virus to spread, whereas otherwise, if it were just coming off the tops of the cells, it wouldn't be able to do that. Now, how do the viruses actually spread? One way is via the blood. We call this hematogenous spread. And when a virus makes a disseminated infection, a systemic infection infecting multiple organs, it may do so by entering the blood. Obviously, the blood can carry it everywhere, so it has opportunity to infect many different organs. The viruses can enter the blood directly through capillaries, or so if a virus gets beyond the basement membrane, it's now in the subepithelial tissues. It can directly enter capillaries. It could be introduced into them by vector bites, of course, and then can get in the blood and spread everywhere. But also, virus in these subepithelial tissues can be taken up by lymphatic capillaries, and that brings them to lymph nodes, and eventually, of course, the lymph system joins with the blood, and so the virus will eventually reach the blood by this route. Lymph capillaries tend to be more permeable than circulatory capillaries, so it's a very good way for viruses to get into the circulation from the subepithelial tissues. So that's hematogenous spread. When you have virus in the blood, it is called viremia. And this is a graph showing a couple of different kinds of viremia. Here we have infected an animal with a virus, and at different times after infection, we are measuring the virus in the blood. So you could just take the blood and do a plaque assay for if the virus forms plaque, or do something else. So here, after you inoculate the animal, you see there's a lot of virus in the blood right away. That's what you put in. That's the virus you injected into the animal. We call that a passive viremia because you get it by injecting virus into the animal. Those titers then go down, and after a couple of days you get another rise in virus titer, and this is from virus replication somewhere in the animal. We don't know where in this particular experiment, but we call it a primary viremia because it is the result of multiplication of the inoculum somewhere. And this is a very typical pattern of virus infections that are spread by the blood. You then have a secondary viremia. So the virus is produced initially by this primary multiplication. They spread to other tissues, and then they seed those tissues and multiply again in those, and then you have a larger burst of secondary viremia. Very typical pattern. Measles virus, for example, does this, and many other viruses as well. So primary is the virus released into the blood after the initial replication at the site of entry. The secondary is produced by disseminated infections. We diagnose virus infections, many virus infections, by the presence of virus in the blood. We don't usually measure virus anymore. We either do antigen tests or PCR-type assays, but we're looking for virus in the blood. And of course, this has a practical consequence that if our blood supply, of course, is donated blood. We haven't figured out yet how to make synthetic blood, so we have to get people to donate blood, and they could have a virus infection of some sort. So we do screen for a number of viruses in the blood supply, including HIV, of course, and HTLV, and all the hepatitis viruses, A, B, C, D, E, and probably G, but I'm not sure. Okay, so we have to get rid of these viruses, otherwise people would get sick. But we don't screen the blood for all viruses. There are viruses in the blood that we don't pay attention to because we're not sure they're a problem. And rather than get everyone annoyed or upset, we just let them stay there. Now here's the pathogenesis of a mouse infection, which was done by Frank Fenner, who just died a couple of months ago. And he was the first to show how these disseminated viral infections develop. And he used a mouse model of smallpox called mousepox. And he showed you inoculate a virus locally, and it does a primary and a secondary viremia, and you get seeding of target organs. So here you inoculate the mouse in the foot pad initially. Here's the virus going in the foot pad. It initially multiplies in the epithelial cells just below where you have inoculated it. Gets below the basement membrane, look, and it's spreading through the regional lymph nodes into the blood. So all those things we talked about, Frank Fenner was essential for us understanding all of those steps. So the mousepox has gone by day 2 into the blood, and that brings it to the spleen and to the liver, and the virus multiplies there. Of course, the bloodstream virus that I'm showing here is the primary viremia because it's multiplied in the skin initially. Then it multiplies in the spleen and liver, and you get a secondary viremia as virus comes out of those organs. And then the virus goes to the skin. So from a foot pad inoculation, it has gone viremic to the spleen and liver. It's causing problems there. It's causing necrosis of those organs. And then it gets to the skin, and that gives you these these early rashes and late severe rashes, which are typical of mousepox. So that virus spread from the foot pad to the skin via these routes. So a typical course of infection. Measles is very similar. You inhale measles. It's respiratory transmitted. Gets into your bloodstream from the respiratory tract and spreads via primary and secondary viremia, and eventually you get a rash, all from inhaling this virus. So these are some viruses that cause rashes in people. Of course, smallpox was the big rash virus infection, but we don't have smallpox anymore. We have eradicated it. It's the only virus infection that we've ever eradicated. But we still have Coxsackie measles, parvoviruses, rubella, varicella zoster that cause various types of rashes. So viruses spreading in the blood. Now we can also spread via neural routes. It's called neural spread. Many viruses spread from the primary site by entering local nerve endings. You have nerve endings everywhere, of course, and why not? Why can't a virus get in it? And for some viruses, this spread is an essential part of the pathogenesis, like rabies, alpha herpes viruses. That is how they spread in you, by going through your nerve fibers. For other viruses, it seems to be an accident. Poliovirus, reovirus, invasion of the CNS is a rare diversion from the normal spread of these viruses, which is typically through the blood. But in a very few cases, in a low percentage of infections, these viruses will enter the CNS and cause problems. And we'll talk about that in more detail as we highlight specific infections later. So here's a diagram of how viruses can spread within the CNS. So here, for example, is a sensory nerve ending. All right, so your skin, of course, is highly innervated. That's why you feel pain under certain conditions. The nerve fibers go back to the CNS. Many viruses can enter sensory nerve endings. They're transported up the nerve fibers by motors all the way up to neurons. This is a motor. This is a muscle, of course, which is innervated eventually by a motor neuron. And viruses can also enter the neuromuscular junction and be transported up the nerve fibers as well. Poliovirus, for example, goes from the neuromuscular junction all the way into the spinal cord to the motor neuron that innervates this muscle. And these, of course, are connected by additional fibers to the primary neurons in the brain. And the viruses can follow these pathways. They can also come out of the CNS into peripheral sites as well. So the travel can be in both directions. Here's a nice picture of the movement of virus in nerves. This is a herpes virus that has been engineered to express green fluorescent protein. And what was done here was to inject the virus in a very specific place in the CNS. And then the virus has spread from neuron to neuron via the axons and dendrites. And you can actually map neuronal circuitry using viruses in this way. You can inject them at very specific places and watch where they connect to and map out the tracings in this way. It's a very interesting application of this reagent. As I said, poliovirus uses neural spread. Polio is a virus you acquire by ingesting the virus, and it goes into your mucosal surfaces in your alimentary tract. It replicates in the mucosa, gets past the basement membrane, and reaches your blood. And in most infections, that's the end of the infection. You get a little fever, perhaps, but the infection stops after about a week. You shed virus in the feces because it's multiplying in this epithelial cells, and you spread it to other people. But for the most part, you're okay. In about 1% of infected individuals, the virus enters the CNS. Probably does so from the motor end plate. The virus gets into your muscle from the circulation, spreads up the nerve fiber all the way to the spinal cord. And then the virus multiplies in the neurons, the motor neurons in the spinal cord, spreads there, and as it destroys them, it causes paralysis. So that's an example of neuronal spread. Now, while we're talking about infections of the central nervous system, let's put up some terminology. Neurotropic virus is a virus that can infect nerve cells. It's all it means. It doesn't imply anything about how the virus gets to the nerve cell. It just means that the virus can infect such cells. A neuroinvasive virus is one that can enter the CNS after you put it at a peripheral site. So if I put a virus into my finger and it gets into my CNS, it is neuroinvasive. If it replicates in the CNS, it's neurotropic. Neuroinvasiveness doesn't mean it's neurotropic. Neuroinvasive just means it can get into the CNS. A neurovirulent virus can cause disease in the CNS. And that's all it means. It doesn't mean that it's neuroinvasive, and it doesn't mean that it is neurotropic, although it should be. So, for example, herpes simplex virus has low neuroinvasiveness, so it's not easy to get in from peripheral sites. But once it's there, it's very neurovirulent. Mumps virus is highly neuroinvasive, but of low neurovirulence. One time before immunization, we are all now immunized against mumps. The measles, mumps, rubella vaccine, MMR, prevents those 3 infections. But before that, kids all got mumps in their young years, and you get the characteristic swelling of your salivary glands. And those kids all recovered. They were fine. But half of those kids, the virus was in their brain, but it didn't cause any problems because it's of low neurovirulence. So the virus could get in your brain. It's neuroinvasive, could replicate, a little bit neurotropic, but not neurovirulent at all. Quite interesting. And finally, we have rabies, which is the worst combination, highly neuroinvasive and highly neurovirulent. So if you are bitten peripherally by a rabid dog, the virus gets into your CNS pretty much 100% of the time, and you're dead. There are very few people who have survived rabies infection without being immunized. There are a couple, though. Okay, so we are spreading by the blood, we are spreading by nerves. Now we have to get into tissues to cause infection of tissues. For example, mousepox infected the liver and the spleen, polio infecting other tissues. How does the virus get from the blood or from the nerves into a tissue in order to multiply? So here we're talking about tissue invasion, and this is largely determined by the structure of the blood vessels that are carrying the virus. So here we have three different configurations of these structures. Here we have an example where we have the blood vessel shown here. You know, capillaries are formed by endothelial cells, and if they're very tightly joined, things can't get through them very easily. So here's the nucleus of this endothelial cell. So it's very tightly joined. Plus there's a basement membrane around it. So here the virus has a lot of trouble getting across. And this is typical in the CNS, connective tissue, skeletal, and cardiac muscle. Very difficult for viruses to cross the capillaries in those tissues. We have another configuration here where there are pores between the endothelial cells, so that lets virus through. There is still a basement membrane, but this is a little more permeable. You find this in the kidney, pancreas, ileum, and the colon. And then in some tissues, the blood vessel is not really a blood vessel. It's just a couple of cells sitting on the walls of the tissue, making what we call a sinusoid. And there's no basement membrane. The endothelial cells are not even joined, so it's very easy for viruses to get through. So that's typical of liver. That's why it's very easy for viruses to get into your liver. So this is a very important determinant of how viruses can get into various tissues in order to multiply. Here's just an illustration of the liver. Here's a sinusoid. It's really just a passage through the liver, so the cells, the hepatocytes, form this passage. And then there may be cells, here we're showing Kupffer cells lining this passage. So viruses that pass through here are readily taken up either into the Kupffer cells or directly into the hepatocytes where they can multiply and spread. Now, not every virus multiplies in the liver, fortunately. Only a few can do that. But it's very easy to get into this organ. The brain is a tough site to get into because it has those very tight endothelial cells and it has the basement membrane, but it is not impossible. So there is a functionally described so-called blood-brain barrier, and this is what makes it difficult for certain drugs to get from the blood into the brain because of these tight junctions. Also makes it difficult for viruses to get through, but some viruses can do it. Not a lot of viruses can get into the brain or the CNS, but there are a few that do. And those routes of entry are illustrated here. You can have, so here is a capillary lumen, so the virus is trying to get from the lumen into the brain matter proper surrounding it. Some viruses actually replicate in the endothelial cells. About that, they bind to receptors, multiply, and get through that way. Some can be taken up by transcytosis without multiplying, so they're taken up into the endocytic system from one side and put on the other. And sometimes if these viruses are present in lymphocytes or monocytes, these cells are very good at squeezing through the capillary endothelial cells, and so they bring the viruses with them. So those cells have adapted to squeeze through here, and so they are like Trojan horses. So that was just a single blood vessel in the CNS. Here's the whole picture, which summarizes all the ways that viruses can get into the CNS. So we have neural spread, right, which can come from peripheral nerves or from the spinal cord, from peripheral muscles. So there are a variety of neural routes of spread of virus into the CNS. And of course, once the virus comes in that way, it's already in the tissue. So it's there into the parenchyma. And here are some examples of various entry routes from blood vessels. So there are meningeal blood vessels, of course. So viruses can cross those blood vessels by the mechanisms we just talked about. And once they're there, they're in the CSF. They can spread to many other places. Viruses can also enter the CSF in the choroid plexus. So that is an organ here that is responsible for producing the cerebrospinal fluid. the choroid plexus is lined with what are called ependymal cells. And some viruses replicate in these ependymal cells and pass through into the CSF. And again, once you're there, you can spread everywhere. And finally, there are cerebral blood vessels through which virus can pass and enter the brain parenchyma. And so, for example, the virus could then replicate in a neuron or an astrocyte or a glial cell. So this is a rare event, virus entering the CNS. But it can be done, and then once the viruses are in there, it's big problems because the CNS isn't particularly good at having a strong immune response to prevent infection. It's almost as if it hasn't evolved to prevent infection very well because it assumes it has put up a physical barrier that would prevent it. Now, an important concept in pathogenesis is tissue tropism. And this simply means the spectrum of tissues that are infected by a given virus. So we talk about enteric viruses, enterotropic viruses. We talk about neurotropic viruses replicating in the nervous system and not elsewhere. Hepatotropic viruses, for example. These viruses have a particular pattern of replication, and that's what we call tropism. So not every virus replicates in every tissue. There's always a subset, and we call that the tropism of the virus. So some viruses have a limited tropism, and others are what we call pantropic. They replicate in many organs. And we would like to know what controls this. What makes a virus replicate in just one tissue and what allows it to replicate in many? Because if we can understand that, maybe we can control the replication in various ways by intervening. So what are the determinants of tissue tropism? Well, one is susceptibility, right? The presence of receptors. If a tissue doesn't have a receptor, it's not going to be infected. So that's a pretty simple one. Then there's permissivity. That is what happens in the cell beyond the entry step. All the proteins that the virus needs to replicate. Intracellular gene products, if they're not present, the cell is not going to be permissive. Accessibility is also important. The virus has to be able to reach the cells that are both permissive and susceptible. It may contact cells that are not, and that doesn't do it any good. And the host defenses. So these all combine to determine tropism. So physical and innate defenses at the site of infection will determine whether the virus is able to replicate there or not. So even if you have cells with receptors that are permissive and can be accessed by the virus, it may not replicate because the defense really is very efficient at preventing infection. Now, going back to the first determinant of tropism, the receptor, there are some cases where the receptor is regulating tropism of viruses, but for the most part, they don't seem to be. So, for example, influenza virus, the receptor is sialic acid, which is on every tissue, yet this virus is largely limited in humans to the respiratory tract. Poliovirus is limited to the alimentary tract and to motor neurons, but the poliovirus receptor is in most organs. So often receptors do not track with the tropism of a virus infection. But other things can control tropism. And here's one example for influenza virus. Now, if you remember the structure of influenza viruses, the viral membrane is studded with glycoproteins. And one of those, the HA, is essential for attaching to host cell receptors. And then it has to catalyze fusion with the endosome as the virus enters the cell. Remember all that? No? Okay. If you recall, the HA has to be cleaved in order to undergo acid-catalyzed fusion. If it's not cleaved, it won't do that because the N-terminal fusion peptide will not be exposed. Cleavage is an essential determinant of flu tropism. So here is a respiratory epithelium which is producing influenza virus. And there is a cell in the epithelium called a Clara cell that produces a protease which cleaves the hemagglutinin of influenza virus. This protease is only made in the respiratory tract. So this virus that's produced is cleaved and it can infect other cells in the respiratory tract. Now let's say some of these influenza viruses somehow get to your liver. There's no protease clara in your liver. So those viruses will infect the liver cell, but then the viruses produced by the liver cell won't have cleaved HA, so they can't infect any other liver cells. So that's why influenza is not hepatotropic, because there's no protease. This is why the virus is restricted to the respiratory tract, because there the protease is produced. So that's one example of how a protease can determine tropism. Now, this is an even broader application for influenza virus, this cleavage of the HA. Some highly virulent avian strains of influenza have a sequence of multiple basic amino acids at the HA cleavage site. So in human strains, the HA cleavage site is only cleaved by this respiratory protease clara. But there are flu viruses that infect birds that wipe out entire flocks of birds. And these guys have multiple basic amino acids at the cleavage site. And what that does, it lets the virus be cleaved by ubiquitous proteases. That is, proteases that are in every cell in the bird. So the flu viruses in birds can replicate everywhere because every cell has the protease to activate the HA. That's why these, in part, are particularly virulent. So the viruses can infect multiple organs of the bird. Back in 1997, avian influenza viruses of this type, we'll talk about what this means a bit later, these were isolated from 16 people in Hong Kong. First time that so many people had been infected with a H5N1 avian influenza virus. So it was a little scary. And these viruses were isolated and sequenced, and they were found to have this basic amino acid insertion at the HA cleavage site. These were very sick people, and the evidence was that the virus had multiplied in multiple organs. So we think that was because this cleavage site now could be activated in multiple organs as well. Interestingly, the 1918 strain, which killed millions and millions of people, does not have these changes. So this is not a pantropic strain. This is just a highly virulent strain. We'll come back to this later when we talk more about influenza virus. Now, shedding of the virus is important because you can only transmit an infection and maintain it in a population if you effectively shed the virus, and it can infect other people. So the virus has to be released from you, from the infected individual, and that is typically required for spread of the virus in a population. The exceptions, of course, are viruses that spread in the germline. So endogenous retroviruses don't have an extracellular phase, or they don't have to, because they're passed from mother to offspring, parent to offspring via the germline. So there's no transmission. Viruses spread in the blood supply, same thing. They're just injected into you, so there's no extra host phase, if you will. But all other viruses have to spread. And this shedding can be at the primary site of replication, where the virus, say, first goes into your lungs, or it can be at distal sites if you have lesions on your skin, which result after spread of the virus from a respiratory infection. That can spread the infection as well. And the number of particles, of course, is crucial for transmission. You have to make a lot to transmit effectively. Respiratory secretions are aerosols produced by coughing and sneezing and speaking, as I have said. Sneezing makes 20,000 droplets. So if you're making influenza virus, each of those droplets is full of influenza virus. And the big drops, so people have studied these droplets very seriously. They measure their sizes. When you sneeze, the big drops fall right near you, the big wet ones. And then the small ones go far. So I could sneeze here, and Saul would be infected in the back of the room with my small droplets, very, very small droplets. So these are the small ones are called droplet nuclei, 1 to 4 microns. They remain suspended, and you inhale them. They can go all the way down into your alveoli and infect the lower tract. And of course, also you can spread respiratory infections by nasal secretions. You touch your nose and you get some virus on your fingers, and you shake hands with someone, or you can even put it on the doorknob. And you know, this room, hundreds of people come in and out of here every day. I would love to sample that doorknob and see what happens. Last year I said I wanted to sample a subway pole for viruses, and the next week one of the students brought me a swab that he used to sample. It's still in my freezer. So here is some sneezing pictures, which I always like to show. Here are the big juicy droplets here, which are going to fall to the ground, and the little ones at the edge of the sneeze are going to spread quite a way. So these are illuminated from behind, so you can see this, but when all of you sneeze, you do this. Coughing makes smaller plumes, but it also makes a plume, and even speaking, and certain words are very good at making plumes, like peas. So all of these things can cause virus shedding. Feces is a major means of spread of viruses in underdeveloped countries, but it still occurs in wealthy nations as well. We routinely contaminate our water with sewage. We have food handlers who are shedding viruses and don't wash their hands and contaminate food and so forth. The blood, of course, is also transmitting viruses. We have vector bites. Healthcare workers get infected. Even though we're careful about the blood supply, there's a lot of blood-borne transmission. Urine also, hantaviruses, are spread from mice to human by mouse urine, HIV, herpes viruses, hepatitis B virus in semen. A mouse virus can even be transmitted to the offspring via milk. Skin lesions will also transmit viruses. Papillomavirus, warts, pox viruses, and even a type of herpes popular among college wrestlers called herpes gladiatorum. You know, these guys are in there rubbing each other, and they transmit this wonderful infection of a herpes virus very efficiently. So as I said, the spread of infection is needed to maintain the chain of infection. We have typically two general patterns of transmission. We have perpetuation in one species, or the alternate infection of insect and vertebrate hosts. So this infection in one species, influenza going back and forth among humans, or a vector-borne disease, a mosquito carrying a virus either among humans or between an animal and a human. Most of the human viruses, it turns out, are transmitted from human to human, most of the ones we know about. So the humans are the reservoir for the virus. Okay? So we are the reservoir for most of our infections. Even influenza, we know a lot of strains originate in birds and in swine, but we are the reservoir when there are outbreaks. We spread it from person to person. And as I said, there are some vector human and vector vertebrate cycles that maintain transmission of the virus. When we talk about a virus going from an animal to a human, we call it a zoonosis, a viral disease shared by humans and animals. So hantavirus pulmonary syndrome, which originated in the four corners of the U.S., came originally from a virus in mice transmitted to humans, and that's a zoonosis because it jumped from the mouse to humans. So here are some examples of transmission routes of different popular viruses. We have the respiratory route, of course, coughing, sneezing, and so forth. Fecal-oral transmission, contact, zoonoses transmitted by vectors of various type, or animal bites, rabies. The blood, many infections transmitted by the blood. Sexual contact from mother to child, including the very important cytomegalovirus and herpes simplex virus. And the retrovirus is transmitted through the germline. These are the endogenous retroviruses. So these are all the ways of transmission that we know about. Typically, where you produce viruses, of course, and their stability is going to determine the route of transmission. So an enteric virus that replicates in the gut obviously is going to be spread by fecal-oral contamination. Envelope viruses tend to be fragile, so they are largely transmitted either by aerosols or by injection or organ transplantation. Non-envelope viruses are a lot more hardy. They can withstand the alimentary tract, high temperatures, and these are often transmitted by also respiratory routes, fecal-oral routes where the envelope viruses in general fail, or fomites, contaminated material. So the contaminated doorknob would be a fomite. And we also have some interesting terms when we talk about transmission of virus infections, actually all infections, iatrogenic, where the activity of a healthcare worker leads to infection of the patient. And included in that is nosocomial infection, where an individual is infected when you're in a hospital or a healthcare facility. Vertical transmission is from parent to offspring. So the transmission of viruses from mother to offspring via the germline is included in that. And all other forms are horizontal transmission. So if you are bitten by a mosquito or you pick up virus from a doorknob, this is horizontal transmission. Vertical is from parent to offspring. And finally, as part of the germline, such as proviral DNA. Interestingly, geography and season also have a large role in regulating patterns of infection. And this is often because the vector or the animal reservoir is geographically restricted in some way. And global travel has really made a big impact on this. Before we had extensive global travel, the spread of viruses or the distribution, I should say, was far more restricted than it is today. With airplane travel being incredibly accessible, the spread of viruses has increased enormously. We'll talk about that in emerging virus infections. And an example of the effect of geography and its effect on the vector is chikungunya virus, how a vector can affect the localization of viral infection. So chikungunya is a member of the togavirus family. It is spread by a mosquito, Aedes aegypti. And when you are infected with this virus, you get typically a rash and joint pains. It was originally prevalent for many years in Asia and Africa, never found in Europe or the United States until 2004. A series of outbreaks spread from Kenya to India, and then a few years later, there was an outbreak in Italy, the first in Europe. The vector, one of the vectors for chikungunya is now another mosquito, the Asian tiger mosquito, Aedes albopictus. So this is the distribution of this mosquito in the U.S. In the year 2000. So it's continuing to spread westward. We still don't have chikungunya virus in the U.S., but the virus has adopted to be able to grow in this mosquito in other places. So previously, this virus only was vectored by Aedes aegypti. As it spread from Africa to India and Europe, the virus underwent a single mutation in one of the glycoproteins, which allowed it to now be vectored by Aedes albopictus. So in Europe, it is often vectored by that mosquito. So eventually, it might reach the U.S. An example of how the initial distribution of the virus was restricted by the vector, Aedes aegypti, and now with its expansion into a new vector, the range of the virus is increasing. Season will also affect virus infections. So what time of year it is in certain climates. Here is rubella from 1963 to 1968, a seasonal peak in the spring. Here is influenza, which of course up here in our climate, in our temperate climates, is a winter infection. Even poliovirus exhibited some seasonality, and this actually varied according to latitude. So you can see it at these latitudes, 35 to 70 degrees, predominantly in September, 25 to 55 degrees latitude, predominantly in March. Why is this? Well, we, for the most part, don't know how virus infections are regulated by the season. But there are some interesting data for influenza virus, which I'd like to tell you about. And these data were acquired using a guinea pig model of transmission, which was actually developed here in New York at Mount Sinai. And what you do is you put guinea pigs in racks. So you have one rack here, you have another rack next to it, and you have a fan blowing air across the racks, right? And you infect the guinea pigs in this rack, and then you look at the transmission to the second rack, and then it transmits very effectively. And what they did in these experiments was to regulate the temperature and the humidity, which of course may be part of the seasonality of infections, the differences in temperature and humidity. And what they found was that low relative humidities were the most efficient or the most favorable for transmission of influenza viruses among these guinea pigs. If you go up to 80% humidity, you don't get transmission among the guinea pigs. So here in the winter, when flu is most prevalent, we have low humidity. Right? It's dry, and you never reach 80% humidity here. So this could, in fact, explain why in temperate climates like ours, there is a prevalence for flu in the wintertime. I also found that transmission in this model was more frequent at lower temperatures, 5 degrees rather than 20. And of course, it's cold and dry here in the humid. And at 30 degrees, which is quite warm, this is centigrade, there's no transmission at all. Yeah?
Any idea why it would be more frequent at 5 degrees? Like, for example, physiological temperature is 37, which is significantly above 30. Why wouldn't it favor a temperature that it could infect that?
Well, here it's probably because the virus, as it's going through the air, it's being desiccated at the high temperature, whereas in the animal, even though it's a higher temperature, it's kept hydrated. Okay, so that's a good hypothesis, but there's some confounding factors that I want to leave you with just to show you that just because the study tells you one thing in guinea pigs, it doesn't necessarily apply to the world. In tropical areas, some tropical areas, influenza is seasonal. Now, tropical areas are hot and humid, right? So why should it be seasonal? If low humidity and low temperature control the seasonality, that doesn't make any sense in tropical areas. In 2009, in April, the pandemic swine origin H1N1 emerged and continued to spread throughout the spring and the summer. I mean, we had flu in July and August of 2009. Makes no sense at all. Totally the wrong season. So the guinea pigs are not making sense here. And so I refer you to this blog post to read more about it. So it looks like low temperature and humidity might explain some of the transmission restricted by weather, but not all of it for sure.